Good morning to all of you. So the last one or two classes that are left will be seeing the good morning once again. So the last 1-2 classes that are left today will be seeing about conversion of units. Basically in physics what happens that even after knowing everything, students commit a blunder, especially while taking units. If you are not thorough with the units, then you can get a wrong answer, and many a times the examiner intentionally will give 4 options. All of them will appear to be similar to each other, only the difference is about their units. So you have to read carefully the question and see what all units they have mentioned. So today we will be discussing about conversion of units and what is the method of doing so. So we will in short introduce something which is known as dimensions. So by that method we will see that how the units of a physical quantity can be converted and what value, how the values will get affected, that also. Again Royal Kitchen Nayan, no, I won't repeat anything. Whatever has been taught, I have told earlier also, whatever has been taught during the online, on that basis just on Sunday or Monday all India level NEET exam you will be appearing for and you will give the NEET, okay, and if you qualify you will become doctor. You need not go to any college also, right? So focus over here. So this is very important. So now conversion of units. Now what happens that whenever we have any physical quantity, like length, mass, energy, force, whatever it be, so that physical quantity will have a definition only when you allot a unit for it, right? For example, suppose I ask you that I have measured something and I got the value as 10. Now can you tell what physical quantity it is? You can't. I told you that I have a physical quantity, I have measured its value to be 10. What is it? You can't tell because I have told only the magnitude, but I did not mention the units. Now if I say that I have measured a physical quantity and it is 10 centimeter, so immediately you can reply that yes, I am talking about length. Or if I say 10 Newton, then you will say that it is what? It is force. So when I attach something which gives you a meaning or which gives the physical quantity a meaning, so that is what we call as an unit. The entity which gives definition to a physical quantity, it defines a physical quantity, it compares two different physical quantities and tells that how different they are from each other. That thing is what we call as a unit. Clear to everyone? So whenever we take a physical quantity, suppose I am taking the physical quantity as x. So this physical quantity x can be represented as Nu. So this x is any particular amount of physical quantity we are taking. So this physical quantity that can be measured, it is represented as Nu. Now this N is called as the magnitude of x in a given system of units, and U is a unit of x. So when we say that a particular unit of x is given, then in that particular system of units, what is the magnitude or the amount or the value that is represented as n. Now suppose I have a given amount of x in my hand, so I will say that if I change the unit, overall the physical quantity is not changing, it is fixed. I have a fixed amount of a physical quantity, say I have sugar in my hand or rice in my hand and it is of a particular value or particular amount. So suppose it is say 1 kg, I am holding 1 kg rice in my hand. If I change the unit from kg to grams, that 1 kg rice, the amount won't change, it will remain the same. But what will happen when I change the unit, the magnitude of the mass of rice, that will change according to the particular unit. So when we write NU, you have to remember that NU, the product, is constant or fixed. N into U is fixed. That means what? That N is inversely proportional to the unit selected or chosen. N is inversely proportional to the unit selected or chosen. So if you have greater unit, that means N will be smaller. So when we write, if unit is large, then N will be small. Unit is large, N will be small. And in two different systems of units, suppose I select in any two different systems of units, I can write, since NU is fixed, so I can write N1U1 equals to N2U2. N1U1 is equals to N2U2. So this applies for every case. So what happens here is, wherever the unit you will see is larger, the corresponding magnitude will be smaller. Magnitude is the value. I will take an example. Listen carefully, you will get answers to everything. Okay. If you listen what I am saying, instead of discussing other very important things like when college will open, or how are you, have you had your breakfast or not, have you taken your lunch or not, all those other important things, if you slightly give a break to them and listen over here, you will get answers to everything, right? N1U1 equals to N2U2. So now what happens here, that what I am saying is, if unit 1 is greater than unit 2, that means I can say that the magnitude of the physical quantity in unit 1 will be less than the magnitude in second unit. Example, what I was just telling, example, if I say X is mass, suppose I am taking the physical quantity X as mass. So say 1 kg. So I can write 1 kg, as I said, will be equal to 1000 grams. Now this kg is one unit, gram is the other unit. In kg, what is the magnitude you are seeing, the value? This is known as the magnitude. So whoever asks the question, listen carefully. This is the magnitude of X, that is mass. This is N1 is 1. In what system of unit? Kg. Now when I take gram, we know the value is this. Now out of kg and gram, you know which one is the larger unit. Kg is larger. How you can check that? You can see, since you can see from here, N2 is greater than N1. N2, 1000 we know is more than 1. 1000 is greater than 1. So this means U1 will be greater than U2. That means kg is a larger unit than gram. So whenever we compare two units, you will have to check whichever system of units has smaller magnitude, that unit will be larger. Is it clear to everyone? Whichever system of units has a larger magnitude will be smaller, and whichever system of units have smaller magnitude will be larger. Okay. So just after noting this particular example, please tell me fast which unit is larger. This is the question, reply fast. Which unit is larger? Meter per second or kilometer per hour? Meter per second is a larger unit or kilometer per hour? Very good. I am getting one reply as kilogram per hour. Janvi, it's not kilogram, it is kilometer. Okay, now we are getting reply as kilometer per hour is the larger unit. Now, what you have done is without thinking you have replied. Now, please try to use this method and then check and see whether your answer is going to change or will it remain the same. Now, all of you replied without thinking. You just gave the reply. Okay. So, the reply that has come from you, kilometer per hour, just I told and you replied. So, this is what mistake generally people commit. Now, the answer is coming meter per second. Let's check fast. If I want to check which is a larger unit, what, can anyone tell me that when you convert kilometer per hour into meter per second, how much is the conversion factor? Kilometer per hour into meter per second. Do you know this? 1 kilometer per hour is 5 by 18 meter per second. Just reply me that do you know this thing or not? 5 by 18 or 18 by 5? What? If you write kilometer, 1 kilometer per hour, let's break this for you. 1 kilometer is 1000 meter and hour is 3600 seconds. So you get this or not? If you divide, 2 zeros gone, 2 5s are 10, 2. Do you get this or not? Clear? Yes. So 1 kilometer per hour, if it is 5 by 18, now tell me from here, this is unit 1 and this is N1, this is N2 and this is unit 2. Which value is more? 1 is more than 5 by 18. So 1 is more than 5 by 18 means N1 is greater than N2. If N1 is greater than N2, so which unit will be more? U2 will be greater than U1. That means meter per second is larger. Which unit is larger? Meter per second. Did you get the answer? So this is what I am telling. In the beginning of the class itself I told that you have to be very careful. These are very basic things. Okay, learning physics, concepts, everything is secondary. First thing, the basics you should have to make it as strong as possible, because if the foundation is weak then the building can't stand. You know it very well. So here meter per second is a larger unit compared to kilometer per hour. So whenever you have the slightest of doubt in your mind, you always have this way of checking. You will just use N1 U1 equals to N2 U2. Is it clear to all of you? Okay. So here whatever I have written, you can quickly take a screenshot so that we move on to a method by which we can convert units of one system to other, and we will see how the magnitude changes. The method will still involve N1 U1 equals to N2 U2, but we will add something in it which is called as dimensions. We won't be going in much detail about dimensions, only the basics and how we will use that along with this N1 U1 equals to N2 U2 to get the conversion of units. Okay. So I hope you have taken a screenshot. So let's move now. So unit conversion by a method of what we call as dimensions. Now what do we mean by dimensions? What we have to do is we have to take any physical quantity and break it into fundamental quantities. So whenever we have a physical quantity x, break into fundamental quantities. Now you all know that what are fundamental quantities? These are those quantities which do not depend on any other physical quantity in a particular way. If they are not dependent on other physical quantities, they have their own independent existence like mass, length and time. Okay, so these are fundamental quantities. Now here, whenever we have a physical quantity or a measurable quantity, first you have to break into fundamental quantities. Example, if x is a mechanical quantity which you are going to study in mechanics, then it can be broken only into three fundamental quantities, that is mass, length and temperature. Understood? What do we call as a mechanical quantity? Mechanical quantities are those which can be broken only into mass, length and time. So if x is a mechanical quantity, then when you break it, so you will see that x will be proportional to mass, it will be proportional to length, it will be proportional to time, and they will have some powers. So mass, length and time raised to some powers. Clear? So such physical quantities are known as mechanical quantities. Now what we do, we write a dimensional formula. When we represent x like this, we call it as a dimensional formula. What we do is basically, we will just see, we will give mass, length and time symbols. Listen carefully, we will give mass, length and time symbols. For mass we will be using m, for length we will be using l, for time we will be using t. But every physical quantity, they may be represented in terms of mass, length and time to some powers which may be different from each other. So I will take the powers as a, b and c. Now some students, they are continuously writing ml, t, a, k. I am talking here strictly about mechanical quantities, so we will restrict our present study only for those quantities which depend on mass, length and time only, not on current or say temperature and all. Okay, so kindly don't repeat the same thing again and again. I have strictly here mentioned that we are now talking about mechanical quantities, so it will depend only mass, length and time. So when we express that x is directly proportional to mass, length and time raised to some powers, then when we write x like this, m to the power a, l to the power b, t to the power c, this thing we call it as something, from now onwards you will call it as dimensional formula of x. So it will be known as dimensional formula. Understood? So now when it is a dimensional formula, in this basis now you can write units of everything. Okay, example, if I have say speed, now how can I write speed as, you know, speed is distance or length by time. So when you are writing length by time, length is capital L, time is capital T, it will go up. Can I write LT minus 1? What about mass? There is no mass, I can write m to the power 0. If I write acceleration, I can write acceleration as v by time. Now I know already for v it is m power 0 L t minus 1 divided by t. So it will become m power 0 L t power minus 2. If I want to write force, I can write mass into acceleration. Now I already know about acceleration, I will multiply with mass. So I will get m L T minus 2. Okay. So these all things we have to remember. If we have, say, energy or work, any form, you can use the formula, any formula that you remember. Energy can be written as force into distance, or work can be written as force into distance. So force we already know, mlt minus 2. One more distance means one more length, so it will become ml2 t minus 2. Just see these examples, you can see from here that whenever we are writing the dimensions, what we are doing, we should know basic formulas, by which we can write the dimensions. Means we have to break all the physical quantities which are given into the simplest form, which are known as fundamental quantities. Here we are talking about mechanical quantities, so only mass, length and time. So on that basis, you can see that speed, acceleration, force, energy, whatever they are, we can always break them into m, l and t, and you have different powers. Is it clear to all of you? Let me know fast. Okay, so once we know this, now if they ask us that convert the units, or from one system convert it into other system, okay, so let's see how we can do that with the help of dimensions. Clear? Okay, so let's proceed. Going by this concept, please take a screenshot fast. So going by this concept, let's see, we have an example. Convert Newton of force into CGS units. Now this is a very simple example, but still we will use the method which I told. Second, if we have a new system, suppose we are taking a new system of units in which unit of mass is 10 milligram, unit of length is 1 kilometer, and unit of time is 20 minutes. Then write (50 joules of energy in the new system of units. So these are two questions that I have. Now the first one you would have directly replied, or you would have found, knowing that SI unit is Newton and CGS is dynes, and 1 Newton is 10 to the power 5 dynes. So first answer so you can immediately give. But the second one, this type of questions you will see later on. These are very easy to do, only just you follow that if you go by the method of dimensions. So let's see how we can do the first one and the second one by the actual method. First what you will do, you see which physical quantity they are talking about. You can see X over here is force. When X is force, so first you write the dimensional formula. We just saw what is the dimensional formula of force, mlt minus 2, mlt minus 2. Now when you write MLT minus 2, now you have to convert 5 Newton of force into CGS units, right? This is done. So what I said, use N1 U1 is equals to N2 U2. Just see the method which I told. N1 U1 equals to N2 U2. Now what you will do, what is N1? They have given 5 Newton. They are saying N2 we have to find. U2 is which unit? CGS units, which is also known as dyne. Clear? So what you will do, and you know that Newton is which units? SI. So what you will just do is just focus on this. This thing you can see is ML, T minus 2 can also be written as T square. So now just fill the values of Newton according to this. When you have mass in SI unit, what is the SI unit of mass? Reply fast. What is the SI unit of mass? Reply fast. SI unit of mass is kg. Yes. SI unit of length is meter. SI unit of time is second. So what you will write in place of Newton? You can write mass, so kg. Length meter. Time SI unit is second square. This you will write here N2. Now come to CGS unit of mass. Mass CGS unit is what? Gram. What is for centimeter? What is here? Second square. So from here second square will cancel. Now they are asking N2. So what will I write? 5 kg gram. Kg can be written as 1000 grams. Meter can be written as 100 centimeter divided by, here also you have gram, here you have centimeter. Centimeter cancels, gram cancels. So you get this as 5 into 10 to the power 5, which I was telling that you could have directly replied this. So 5 into 10 to the power 5. So 5 Newton of force is equals to 5 into 10 to the power 5 CGS units of force. Understood this thing? So this is what we do. Now this was a simple question where we directly know that yes, we have 1 Newton has 10 to the power 5 dyne, so we can answer. But sometimes you will see that they change the system of units and they can mix and ask anything. So on that basis, how we will find? The method is same. Let's quickly see the next question, which will make you clear with the method which I just said. If you see the second question, if you see, they are talking of 50 joules of energy, means here X is energy. Now I just told, if you have energy, this means the dimensional formula of X is energy is force into distance, ml2 t minus 2, ml2 t minus 2, which is nothing but ml2 by t square. Now again use N1 U1 is equals to N2 U2. What is N1? 50 joules. N2 we have to find, we have to convert 50 joules in new system. So what is U2? This is new units. Now joule we know is SI. Joule is SI unit. We have to find N2. So how we will proceed? Let's see. So now what we can write from here is 50 joule. Just see the unit, ml square by t square. So SI unit of mass, kg. SI unit of length, meter, but we have square. SI unit of time, second, but we have square. If you go to the right side, we have to find N2. Keep it like this. Now you have new units. Again you have to write mass. What is the new unit of mass? Read the question. They have given 10 milligram. 10 milligram. So 10 milligram. Then we have length square. What is the unit of length? 1 kilometer. So length square, 1 kilometer square. What is the unit of time? Time they have given is 20 minutes, but I have to take time square. So I will write 20 minute square. Now final step, break them into simpler forms. So now what we can say from here is 50 kg, and here we have milligram. Kg you can convert into 1000 grams. Meter you can convert, here we have kilometers, so take it meter only. So we have meter square divided by second square. Keep it as it is. Now after keeping it as it is on the left side, see the right side. We have N2. Now 10 milligram. Milligram means how much? Milli means 10 to the power minus 3 grams. Kilometer, break into meters. We have 1000, that is 10 to the power 3 meter. And below you have minute, break into seconds. So 20 into 60 seconds whole square. Now you can cancel all this gram, meter, second, both sides. So finally you get the value of N2. Just cross multiply. You will have 50 into 20 into 60 whole square into 1000 divided by, you can see 10 to the power minus 3 and 10 to the power 3 whole square, that is 10 to the power 6. So from this case you can get the value. So just to tell you that these type of questions are very common, you are going to see such questions. So what you have to do, you just focus on one thing, that first you have to write that which physical quantity is taken under consideration. Like in the previous example it was force, now it is energy. Second thing, after writing that particular physical quantity, write its dimensional formula. So now using finally N1 U1 equals to N2 U2, so you know one unit, you know its value. In second unit you have to find the value or magnitude. So writing in this way and breaking units into simpler forms like kg into gram, milligram into gram, meter is already in meter only, kilometer can be converted to meter. Second we already have here, minute you can convert into second. So after writing these, whatever is unknown you will get it. Clear? So this is the method that we use in conversion of units. So I hope this concept is clear to all of you. You can go through whatever we studied just now. So that will be all for today's class. Okay, thank you.
Good morning, students. So today there is a new lecture 4. We have to count total number of structural isomers. So now there will be complete application of which we have studied in these 2 or 3 lectures about all the structural isomers. So that we have to remember isomers of alkyl and different type of structural isomers. So today we are only going to discuss about the questions based on structural isomers. So let us start with first question. You all have to give answers for this. Find minimum number of carbon atoms required. Number of carbon atoms required to show position isomerism. So special type of structural, but we are asking only position isomerism. So if any compound has to exhibit position isomerism, how many minimum carbon atoms are required? Apply your mind. Apply definition of this position isomerism. Same length of parent chain but different position of substituent. So all students are writing 3. So in first thought, always we will think that 3 carbon atoms are required. So it is not bad that you are thinking 3 carbon atom. So what you are thinking that if we take 3 carbon atom, let us say chlorine at first position, 3 carbon atom chlorine is at second position. So what we are thinking, these are which type of isomer? Position isomer. But in question, have they said that only one substituent we have to take? It can be substituent, 2 substituent also. So if we take 2 substituent, then how many carbon atoms are required? Only 2 carbon atoms are required. Now if we take this structure, now what is position of substituent? Position of substituent is 1, 1. And if I take carbon, carbon, chlorine, chlorine, now what is position of substituent? 1, 2. So if you check here, how many carbon atoms are present? Only 2 carbon atom. So in first thought, we will think always that how many minimum carbon atoms are required? 3 carbon atoms are required. But that is only in that case where one substituent is present. But here it is not given in question that we have to take only one substituent. So what is answer for this question? How many minimum carbon atoms are present? 2 carbon atom. So remember this, answer will be 2 carbon atoms. If I modify this question that how many minimum carbon atoms are required to show position isomerism having only one substituent, then answer will be 3 carbon atom. But if they ask only to show position isomerism, then what is answer? 2 carbon atoms. Clear? Any doubt in this 2 carbon atom? Okay. So let us move to next question. Now before moving to next question, just I am asking you some questions which we have written earlier. Isomers of alkyl. Sequence you have to tell. C3H7, C4H9, C5H11, C6H13. What is sequence for this isomers of alkyl group? 2, 4, 8, 17. Very good. So for 3 carbon atom, how many isomer? 2. 4 carbon atom, 4. 5 carbon atom, 8. 6 carbon atom, 17. So these are structural isomers of alkyl group. Now if we know these, how questions will be very easy. If they ask calculate total number of structural isomers of C5H11Cl. If this question is given that we have to find total structural isomers of C5H11Cl. Now if we do not know these isomers of alkyl, then what we do, we'll draw all the possible structures for C5H11Cl. First we find degree of unsaturation, so it will take time. But now directly we know that how many isomers of C5H11 are possible? 8. So direct answer is how much isomers are possible? 8 isomers. So what we write this as C5H11 and what is present? Cl. And for C5H11 how many structural isomers are there? 8 structural isomers are there. So this is answer. So in one step we can tell answer. So there is no need to draw the all possible isomer, then we have to count. So this direct approach we can apply. So that is only possible if you remember this sequence 2, 4, 8 and 17. Any doubt in this question? Clear? Now let us move to next question. Now we'll do different type of functional groups. First functional group will start with alcohol. Then we start with ether. Then combine alcohol and ether. Okay. So first question is calculate total number of structural isomers of alcohol having molecular formula C4H10O. But we have to find total number of structural isomers of alcohol whose molecular formula is given how much? C4H10O. Okay. Now we know how to solve these questions. Which functional group is alcohol? Alcohol functional group is ROH. We all know that OH functional group is called as alcohol functional group. Now in this formula C4H10O, can we represent this as C4H9 and what is present here? OH. C4H10O can be written as C4H9 and what is present here? OH group. And how many isomers of C4H9 are possible? 4 structural isomers are possible. So what is answer for this question? How many structural isomers are possible? 4 structural isomers are possible. Okay. So whenever these type of questions will be asked, there is no need to draw structure. Directly we can mark answer OH separate. What is left? C4H9. And for C4H9, how many isomers are present? 4. So direct answer is 4. No need to draw the structures. Okay. But next point is, here they are not asking how many primary alcohol, how many secondary alcohol, how many tertiary alcohol. So what do you mean by degree of alcohol? We will tell about what is meaning of degree of alcohol. You have to tell about what is meaning of degree of alcohol. Same like degree of amine is there, degree of amide. So you have to tell what is degree of alcohol. Degree of carbon attached to OH. Very good. So degree of alcohol is degree of carbon directly attached to which group? OH group. Now carbon can be primary, secondary and tertiary. So alcohol is also primary alcohol, secondary alcohol, tertiary alcohol. Now representation is very important. So first I am writing representation for which alcohol? Primary alcohol. So how to write primary alcohol? RCH2OH. Now if you check, this OH is directly attached to this carbon. How many hydrogen are present? 2 and 1 carbon is attached. So this carbon is primary carbon. So that's why alcohol is called as which alcohol? Primary alcohol. Clear? Next is secondary alcohol. How to write secondary alcohol? We have to attach OH to that carbon atom which is secondary carbon. So how to write R, OH and R. Now carbon attached with 1 hydrogen on both sides, which group is present? Alkyl group. So that's why this is representation for which alcohol? Secondary alcohol. And if we write representation for tertiary alcohol, OH will be attached to which carbon? Tertiary carbon. So with carbon, How many alkyl group we have to attach? 3 alkyl group. And what is attached here? OH group. So OH is directly attached to this carbon, and how many carbon are directly attached here? 3. So this is tertiary carbon. So alcohol is called as tertiary alcohol. So this representation is very important for primary, for secondary, and for tertiary. Now a difficult type of question can be asked. If they ask total isomers of alcohol, it is very easy. We'll write OH, rest alkyl group, we'll write isomers for that. Same in case of, we have written for last one, C4H9OH. But if they ask individual, that how many primary are present, how many secondary are present, how many tertiary alcohol are present, then one way is that we'll write all the isomer, then identify which is primary, which is secondary, which is tertiary. It will take time. But directly, if we remember this representation, we can find easily how many are primary, how many are secondary, how many are tertiary. So let us take one question. Question is calculate total number, or will not write total, calculate number of primary, secondary and tertiary alcohol having molecular formula C5H12O, and what we have to count? Only structural, not stereo, only what we are writing structural. Now we have to find out differently how many are primary, how many are secondary, how many are tertiary. Okay. So let us start with primary alcohol. Now I want to know from all you students how it is possible, how you are multitasking. It means you are not listening. Everyone is busy in messages. So I will tell you, students who are busy in messages, they require many long-term attempts for NEET. And students who listen very carefully in class. So there is one term. Selection is always directly proportional to sincerity. That how much sincere you are. If you are not sincere, then leave this. Selection is not for you. Selection is only for those students who are very sincere. And similar type of atmosphere you will create in that also, offline classes also. And when you come in offline, you will not make a single noise. You will be beaten in classes. So take advantage of these online classes. Talking stupid in chat, unnecessary. Important topic is going on. Now let us first start with primary alcohol. And students who do not want to study, they can leave the class. Go to some other place. Why wasting time? Now primary alcohol, what is representation? R CH2OH. It means what we have to fix? CH2OH is a fixed compound. After that alkyl group is present. So if we add here CH2OH, after CH2OH, one carbon atom is removed. So 5 are total present. So how many carbon atoms are left? 4. So what is here? C4H9. And for C4H9, how many isomers are possible? 4 structural isomers are possible. So in question, if they ask how many primary alcohol are present, so how many primary are present? 4 primary alcohols are present. Clear? Next, if we talk about secondary alcohol. Now for secondary alcohol, what is representation? R, CH, OH and R. Now on both sides we have to add alkyl group. So let us start from simplest alkyl group here, CH3. CH, what is present? OH. Now how many carbon we have fixed there? 2 carbon. Total are how many carbon? 5. 2 are fixed, so remaining how many carbon are present? 3. Now for C3H7, how many isomers are possible? 2 isomers are possible. Now next case is, it is not necessary that only CH3 can be present here. We can write C2H5 also. And here CHOH. So here also what is present? C2H5, because total carbon atoms are 5. 2 plus 2, 4. 4 plus 1, 5. In C2H5 there is no isomerism possible. So only this one structure is possible. So how many secondary alcohol are possible? 2 plus 1, 3. So if they are secondary alcohol, so for secondary alcohols, how many structural isomers are possible? 3 structural isomers are possible. Next, if we move to tertiary alcohol. Now in tertiary alcohol, what is the representation? With carbon, all alkyl groups we have to take, and one is OH. So one carbon is fixed, remaining 4 carbon atom we have to distribute in alkyl groups. So what are possible cases? One methyl, another is also methyl. So again, what is left? Ethyl. So 2, 3, 4, 5, all carbon atoms are completed. Here what is present? OH. Now if you check, there is no isomerism possible here. So how many structure are possible? Only one structure is possible. So if there's total number of isomers, so 4 plus 3, 7, 7 plus 1, 8. So 8 directly we can write C5H11OH, and for C5H11, how many isomers are possible? 8 isomers are possible. But if there's individual, how many primary, how many secondary, how many tertiary? So primary are 4, secondary are 3, and tertiary are 1. So total answer will be 4 plus 3, 7, 7 plus 1, 8. Clear? Now this is about representation of alcohol, then only we can find primary, secondary and tertiary. So 4, 3, 1. So this is about alcohol. So in alcohol only 2 types of question will be asked. One is total number of alcohol and one is primary, secondary and tertiary. Now let us move to next, ether functional group. Now ether and alcohol are functional isomer. So for same molecular formula, alcohol is also possible, ether is also possible. Now I will write combined question, that find total number of structural isomers of alcohol and ether having molecular formula C4H10O. Alcohol also we have to find and ether we also have to find. So first you have to tell about alcohol, because alcohol we have discussed earlier. Then we move to ether part. So first you have to tell about how many alcohols are possible. Very good. Alcohols we have studied in last to last question. How many are possible? 4. So there is no difficulty in writing how many alcohols are possible. Just what we have to write, C4H9, and which group is present here? OH group. And for C4H9, how many isomers are possible? 4 structural isomers are possible. Yeah, so it is easier. But if we have to write about ether, now which is ether functional group? Ether is ROR. Now on both side of oxygen, alkyl group we have to write. So let us start with first simplest alkyl group, what is present? CH3. Oxygen. Total 4 carbon atoms are present. 1 we have written, so remaining are 3 carbon atoms, C3H7. And for C3H7, how many isomers are possible? 2 structural isomers are possible for C3H7. Clear? Next case, what we can do here, in place of CH3, what we can write? C2H5. Oxygen, here also what is possible? C2H5. So in C2H5, there is no possible isomerism. It means only this is one structure. So 2 plus 1, 3. Now do not do like this, that C3H7, CH3, that will be identical. That on oxygen, one group is methyl, one group is propyl. If you write here propyl, methyl, that will be same. So how many are possible? 2 plus 1, 3. So 3 structural isomers for ether are possible. So what is answer for total number of isomers? 4 are alcohol, 3 are ether. So total answer will be 7. Okay, so what is total answer? 7. 4 are alcohols and 3 are ether. So total number of structural isomers are 7. Okay. Now this is for alcohol ether combined. Now next question I will give you, only you have to find for ethers, that how many ethers are possible for a molecular formula. Okay. So write down question. Find total number of structural isomers of ether having molecular formula C5H12O. Solve this question. Now I am not asking alcohol, only I am asking ether. How many ethers are possible with this formula? Structural isomers. Three are possible only. Try to solve. Take all possible cases on both sides. How many alkyl groups you can write? Somebody is sending 6 also. Now 3 is not possible. No? So representation is ROR. So let us start from first group CH3. Now one carbon we have taken. How many are left? 4. C4H9. And for C4H9, how many isomers are possible? 4 are possible for C4H9. So 4 are already here. Now next case. In case of CH3, what we can take? C2H5 oxygen. Now 2 carbon are present, total are 5. So how many are remaining? 3. C3H7. So for C3H7, how many isomers are possible? 2 isomers are possible. Next, if you take C3H7, C2H5, identical. Next here C4H9, CH3, identical. So total how many are possible? 4 plus 2, 6. So how many total isomers are possible for this? Total ether are 4 plus 2, 6. Maximum students have given answer 6. Very good. Any doubt in this question? Now why these questions are looking like simple question? Because we know isomers of alkyl. If you do not know isomers of alkyl, then these questions will be very difficult for you, because you have to draw all the possible structure. Then you cannot draw these all possible structures. Clear? Now when we are studying about functional isomers in last class, I have written representation for primary amine, secondary amine and tertiary amine. How many student remembers about representation of amines we have written? Primary, secondary, tertiary. Remember amines we have written? Primary amine, secondary amine and tertiary amine. So I'll write again. If we have to write representation for primary amine, how to write? RNH2. This is representation for which type of amine? Primary amine. If we have to write representation for secondary amine, then it is RNHR. On both sides, what is present? Alkyl group with NH group. And in tertiary amine, what is representation? N. And on all sides, what is present? Alkyl group. So what is important? That in case of primary amine, what we have to fix? NH2 group. In case of secondary amine, what is fixed? NH group. And in case of tertiary amine, what is fixed? Nitrogen. Here 3 alkyl groups are present. Here 2 alkyl group. Here only how many alkyl group? 1. So whenever they ask question from total number of isomers of amines, so for total isomers of amine, we have to find out separately how many primary are possible, how many secondary are possible, how many tertiary are possible. Then we can calculate total number of isomers, otherwise we cannot calculate. So if I write a question that calculate total number of structural isomers of amines having molecular formula C4H11N. Now one question I will solve for you. Next question I will give that you have to solve. So let us start with first primary amine, because if they are writing amines, for total we have to find out primary, secondary as well as tertiary. So let us start with first primary amine. Now for primary amine, what is representation? It is RNH2. What is representation for this? RNH2. Now if we separate NH2 from here, what is left? C4H9. And for C4H9, how many isomers are possible? 4 structural isomers are possible. So it means how many primary are possible? 4. Easily we can find out. Now let us move to secondary amine. So if we have to write secondary amine, what is representation? R, N, H, R. On both sides we have to write alkyl group. So let us start with first simpler alkyl group is CH3. NH. Now total carbon atoms are 4, 1 we have fixed, remaining carbon atoms are 3. So for C3H7, how many isomers are possible? 2 structural isomers are possible. Next, if we take case in place of CH3, what we can take? C2H5. NH, here also what is present? C2H5. So there is only one possible case for this. Now next is not possible. If you write C3H7, CH3, that will be identical. So how many secondary amine are possible? 2 plus 1. 3 structural isomers are possible for secondary amine. Now let us move to tertiary amine. So if we write tertiary amine, so nitrogen attached with all what we have to write? Alkyl group. Now total 4 carbon atoms are present and we have to distribute in 3 alkyl group. So what are possible case? CH3, nitrogen, CH3, and how many carbon are left? Only 2, C2H5. So how many structure are possible here? Only one structure is possible. So primary are 4, secondary are 3, tertiary is 1. So what is answer? Total is 8. 4 plus 3, 7, 7 plus 1, 8. So if we write how many total isomers are possible? 4 plus 3 plus 1. Total 8 structural isomers are possible for this compound. Amines. Having which molecular formula? C4H11N. Okay. So in case of amine, you have to find separately. First primary amine, then secondary amine, then tertiary amine. Any doubt in this 8 isomers, primary, secondary, tertiary? Ask any doubt in this. Otherwise, I will give next question which you have to solve for amines. No doubt. Let us write one question. Separately, you have to tell primary, secondary, and tertiary. Do not tell direct answer that how many are total. In this form, you have to send answer 4 plus 3 plus 1. This type of answer. So question is find total number of structural isomers of amine having molecular formula C5H30N. Now take your time, 1 minute or 2 minutes, but you have to send answer in that form. Primary plus secondary plus tertiary. That how many are primary, how many are secondary, how many are tertiary. Do not send only answer for total. Okay. Try to solve this. First you have to send answer for primary. Somebody is sending answer for primary. Correct. Primary are 8. Very good. Then move to secondary. Somebody is saying 8, 7, 3. No, 18 is not correct. Only structural we have to count. So primary are 8, that is correct. Now move to next secondary. How many secondary are possible? Secondary are possible. What you are writing? Some students are writing 6, some are writing 4. Now secondary are 6. Secondary are 6. Very good. Now try to find tertiary. How many tertiary are possible? All subject 8, 6, 2. Try to work on tertiary. In tertiary there are not 2 isomers. In tertiary, how many are possible? 3 are possible. Tertiary, how many? Possible? 3. So, total answer, what you are getting? 8 plus 6 plus 3. 8 are primary, 6 are secondary and 3 are tertiary. Total 17. 1 or 2 students are telling correct. 8 plus 6 plus 3. Very good. Total how many isomers are there? 70. Now let us start. Now this is important question. Now this is in NCERT also. In second year NCERT, you will find this type of question that total isomers of C4H11N, C5H13N. So how to draw or find out these isomers? So let us start with primary amine. Now there is no difficulty in finding primary amine. Just we have to write NH2. So what is left here? C5H11. And for C5H11, how many isomers are possible? 8 structural isomers are possible. So there is nothing difficulty in finding how many primary amines are present. So primary amines are how many? 8. Now let us move to secondary amine. Now for secondary amine, what representation we have to write? R NHR. So we have to start from simplest alkyl possible. What is simplest alkyl? CH3. Next is NH. Total 5 carbon atoms are present. 1 we have already written. So how many are left? 4. So C4H9. And for C4H9, how many isomers are possible? 4 structural isomers are possible for C4H9. Next case we can take in place of CH3, what we can write? C2H5 NH. Then here what is left? C3H7. And for C3H7, how many isomers are possible? 2 isomers are possible. So 4 plus 2, 6. Next if we write CH3, C2H5, same it will be C3H7, C2H5. Next C4H9, CH3, that will be same case. So how many secondary are possible? 4 plus 2, 6. Let us next move to tertiary amine. Now what is representation for tertiary amine? It is R, N, R and R. So this is representation for which type of amine? Tertiary amine. Now total 5 carbon atoms are present. First case we can take, we'll write both methyl group. So 2 carbon atoms are completed, CH3, CH3. Next total 5 are present. So how many are remaining? 3. C3H7. And for C3H7, how many isomers are possible? 2 isomers are possible. Now it is not necessary that both are methyl. If we add 1 methyl, 1 ethyl, CH3N. Let us take 1 is ethyl. Then what is left? 2 plus 3 carbon atom. So 2 are left. So it is also C2H5. So in C2H5 there is no isomerism. So this is only one structure. So how many secondary amine are there total? 6. How many tertiary amine are possible? 2 plus 1, 3. And how many primary are possible? 8. So if we write total number of isomers what we write 8 plus 6 plus 3. So what is answer for this question? How many total isomers are possible? 17 isomers are possible. Check any doubt in this? Total 17 isomers for C5H13N. Okay. So what we have discussed today, how many functional groups we have covered? First functional group what we have covered, alcohol. Second functional group what we have covered, ether. Then combined question of alcohol and ether. And in alcohol one case question will be separately primary, secondary and tertiary. After that, if two questions are based on amine, for amine what is important, their representation, primary, secondary and tertiary. Same is important representation for primary alcohol, secondary alcohol and tertiary alcohol. Clear? So I hope this is very easy for you if you remember how many isomers of alkyl are possible and what are their representation. So in next class what we discuss, carboxylic acid, ester, and one thing what we discuss, amide also. So we have written representation for amide also, so that you have to read before next class. Then I think tomorrow will be last class of this online session. So in last class what we cover, amide, carboxylic acid and ester. Okay. So students, thank you for today.
Hello, good morning, students. Good morning. Good morning, everyone. Good morning. Busy among yourself? Yeah, Charita, good morning. Kedari, good morning. Good morning. Yes. Yeah, I'm fine, Tikshana. I'm good. Good morning. Good morning, everyone. Okay. Yeah, I'm fine. I'm fine. I'm fine. Okay, did you all read yesterday's topic? Did you all read? Yeah, I'm good. I'm good. Did you all read? Still Lahari only. Okay. Yes. So quick questions, 2 questions, and after that 2 or 3 questions. After that, yeah, we'll go to the today's topic, that is meiosis. Okay, in mitosis, okay, how many stages are there in karyokinesis? In mitosis, how many stages are there in karyokinesis? Karyokinesis. 4 are there. Very good. That 4 are. Can you name them? 4. Correct. 4 is correct. Correct. Name them in the sequence. Prophase, anaphase, telophase, metaphase. What is the sequence? Prophase, metaphase, yes, Madhumita. Prophase, metaphase, anaphase and telophase. Correct. Okay. Prophase, metaphase, anaphase and telophase. Very good. What do you mean by karyokinesis? What do you mean by the word karyokinesis? Karyokinesis. Yeah, karyokinesis meaning. Is it cytoplasmic division or division of nucleus? Yeah, Deeptika, very good. Division of nucleus. Yes. Sairaj, yes. Tikshana, yes. Nuclear division. Among these 4, prophase, metaphase, anaphase and telophase, which is the longest phase? Nuclear division, very good. Which is the longest phase? Prophase, metaphase, anaphase and telophase, which is the longest? Prophase, very good. Very good. Correct. By the end of the mitosis from one parent cell, how many daughter cells are obtained? By the end of the mitosis, how many daughter cells are obtained? 1, 2, 3, 4. Which is correct? How many daughter cells are obtained? Very good, 2. Who said 4? Not 4 ma, 2. Guys, it is 2. 2 daughter cells are formed. What is another name of mitosis? Another name of mitosis. Is it reductional division or equational division? Which is correct? What is another name of mitosis? Reductional division or equational division? Yes, Afifa, equational division. Okay. Yeah, equational division. It is another name. Very good. Okay. So now, let us proceed to the today's topic, that is meiosis. Okay. Meiosis, it is also called as reductional division. Meiosis is also called as reductional division. And in this one parent cell, it occurs always in the diploid cells only. And generally it occurs in the reproductive cells. Whereas mitosis will occur in the somatic cells. So reproductive cells or germ cells, they will undergo the meiosis. And one parent cell, this is called as one parent cell, it will undergo meiosis and it will be forming finally, four daughter cells. So, four daughter cells are obtained by meiosis. So, in the meiosis, which is actually called as reductional division, one parent cell will be undergoing the meiosis, and after the complete meiosis, we will be getting four daughter cells. I have written like stepwise manner. This first one is called as meiosis I, and this one is called as meiosis II. So, it occurs by two steps or two stages, which is called as meiosis I and meiosis II. And in this meiosis I, so we can say that meiosis is divided into meiosis I and meiosis II. But in between these two, we have one more stage which is called interkinesis. Interkinesis. So, meiosis is divided into meiosis I and meiosis II. In between, we have the interkinesis, where the cell will be preparing itself for undergoing the division, but there is no DNA replication here, no DNA replication. So, first meiosis I. Again, this meiosis I is further subdivided into karyokinesis I and cytokinesis I. K for karyokinesis and C for cytokinesis. So, karyokinesis I and cytokinesis I. So, in meiosis basically 2 times nuclear division and 2 times cytoplasmic division will happen. So, karyokinesis I and cytokinesis I are part of meiosis I. In the same way, meiosis II also will be having the same karyokinesis II, cytokinesis II. This we will see anyhow tomorrow. Now, let us concentrate on this. Karyokinesis I is further subdivided into 4 phases: prophase I, metaphase I, anaphase I, telophase I. You know all the same things: prophase, metaphase, anaphase and telophase. PMAT. So, these are all one because we are taking the karyokinesis I. So, what will happen in meiosis I means one parent cell, which is actually diploid cell, it will form, by the end of this, it will form 2 daughter cells, which are haploid. So, that is the reason why meiosis I is actually called as reductional division. Reductional division. Though I have given the nickname of meiosis is reductional division, the actual reduction will happen in the meiosis I. So, meiosis I is actually called as reductional division. Whereas meiosis II, it is similar to, it is exactly similar to mitosis, which is always the equational division. So, it is also called as equational division. So, like this, the meiosis is divided into 1 and 2. 1 is further divided into karyokinesis I, cytokinesis I. Karyokinesis I is further divided into prophase I, metaphase I, anaphase I, telophase I. After that, cytokinesis will happen one time, and it results in the formation of one diploid parent cell will be forming 2 haploid daughter cells. So, as the ploidy got reduced to half, the number 2n got reduced to n, it is called as reductional division. In meiosis II also, you have karyokinesis II and cytokinesis II, and that is exactly similar to mitosis. So, meiosis II is called equational division, but on the whole, meiosis is called as reductional division. This is how the meiosis will happen. Okay, I hope all of you understood. Yes, ma? Have all understood? Guys, okay. Okay, meiosis is divided into how many stages? Meiosis. Meiosis is divided into how many stages? 2 stages. What are those 2? 4? No, not 4, 2. Which is the first one? Which is the first one? All of you see the tab here and tell me which is the first one. Yeah, good. Meiosis I and meiosis II. Very good. Meiosis I and meiosis II. There is one phase in between meiosis I and meiosis II. What is that? What is the stage that is present between meiosis I and meiosis II? Interkinesis. Very good, Ananya. Very good. Interkinesis. Okay. Meiosis I is further divided into karyokinesis 1 and cytokinesis 1. Okay, interkinesis. Among meiosis I and meiosis II, which is called reductional division. Among meiosis I and meiosis II, which is reductional division. 1. Very good, Tikshana. M1. Yes. Meiosis I. Good, good, good. Which is similar to mitosis, or it is also called equational division. Similar to mitosis, or it is also called as equational division. Which one? Only meiosis II, not meiosis I. It is only meiosis II. Okay. Now all of you can see here the diagram. Interphase will be like this. The interphase cell is looking like that. It is undergoing meiosis I and meiosis II. In meiosis I, this is the karyokinesis 1. Okay, after this, cytokinesis 1 will happen. And after this, cytokinesis 2 is happening. So, meiosis I, it is divided into, karyokinesis 1 is divided into prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2, again karyokinesis 2 is divided into prophase 2, metaphase 2, anaphase 2, telophase 2. Cytokinesis 1, cytokinesis 2. So, by the end of this one, you will be getting 2 daughter cells. And by the end of the meiosis 2, you will be getting 4 daughter cells. This is exactly the meiosis, what is going to happen. In between here, we have the interkinesis, where there is no DNA replication, just the cell will be preparing itself. Okay, there is no particular DNA replication. It will take some rest and it will continue the meiosis 2. Okay. Now you know what is the first stage in the meiosis 1. What is the first stage in the karyokinesis 1? In meiosis 1, karyokinesis 1, which is the first stage? Karyokinesis 1, which is the first stage? Prophase 1. Yeah, you have to use the numbers. Prophase 1. When you simply write prophase, it belongs to mitosis. It is prophase 1. This is the first stage. Then metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. Okay. But out of all these, prophase is complicated and it is longest. Longest and complex process. Prophase 1 is longest and it is the complex stage. Anaphase is generally shortest phase. Anaphase is shortest and prophase 1 is the longest. Okay. Is meiosis a part of cell cycle? Yeah, it depends on the cell. If it is a reproductive cell, yeah, obviously it is a part of the cell cycle. The cell will be going either into the, yeah, Venkata Sivakumar, yeah. The cell will be going into the mitosis or meiosis. It depends on the cell type. Yeah, correct. Venkata Sivakumar, yeah. Leptotene, zygotine, pachytene, diploatine and diakinesis. Very good, very good. You read already. You know. Good. So, the longest phase is the prophase 1 and the shortest phase is the anaphase 1. Or generally anaphase is shortest and longest is the prophase. Now, let us see in prophase. Prophase, where all the changes of prophase of mitosis you have learnt, like condensation of the chromatin will happen. so that it will become the chromosomes. Then after that, the nucleolus, nuclear envelope, okay, then ER, Golgi, they disappear. The two centrioles pair, they'll be moving towards opposite poles in animal cells, and asters are formed, okay, and that asters will be giving rise to the spindle fibers. These are the events which will happen throughout the prophase 1 also. But this prophase 1, as I said, it is complicated and it is the longest phase. It is subdivided into 5 phases. Only the prophase 1 is subdivided. Prophase 1 is subdivided into leptotene, zygotene, pachytene, pachytene or pachytene, diplotene, and diakinesis. So prophase 1 is subdivided into 5 phases. You have to remember the order also. LZP D dia, first letters. The sequence also will be asked in the NEET. Okay. So LZP D dia, because diplotene is there. So you can't write DD. So leptotene, zygotene, pachytene, diplotene, and diakinesis. This is the sequence how they will happen in that prophase 1. So the first phase in the prophase 1 is leptotene. Let us see what are the changes that will happen here. Already I told you condensation of chromatin will begin here and it will be continued throughout all the phases. Condensation of chromatin. Occurs throughout prophase 1. From starting leptotene to diakinesis, the condensation of chromatin will be going on. Compaction of chromatin will be going on to become the chromosomes. That will be starting in the leptotene and it will end by diakinesis. In the diakinesis, there are other changes which will happen, the final stages of the prophase events. So what are the major changes that will happen in these 5? Let us see the first prophase 1, later we will proceed to the metaphase 1, anaphase 1 and telophase 1. So what are the subphases of prophase 1? Subphases of prophase 1. Guys, focus. What are the subphases of prophase 1? Yes, Venkat Sivakumari, you already told, very good. Leptotene, zygotene, pachytene, diplotene, diakinesis. Yes. Only one. Others? Guys, 5, very good, Lekhya, 5. Okay. Venkata Nageswarao, leptotene, LZP, D dia, very good. You can remember the sequence. Vaishnavi, yeah. Very good, Vaishnavi. Little Zera, please do dance. Wow. Good. Yeah, after that mnemonic also, please do remember the stages names, okay? Leptotene, zygotene, pachytene, diplotene and diakinesis. Very good, very good. Okay, correct. Now let us proceed to the events of this prophase 1, how it happens. All of you can see? Yeah, very good. Yeah, LZP, D dia, very good. So now coming to the meiosis prophase 1 stages. The first one you can see, this is the leptotene. You know guys, this time in the NEET paper about leptotene it has come. The question has come about leptotene. Okay. So in the leptotene, the chromosome condensation will begin. They start undergoing the compaction and they gradually visible under the electron microscope. We can see them. Okay, the condensation begins there and we can see the chromosomes, they gradually visible in the leptotene. That is why you can see here that chromosomes are gradually appearing in the leptotene. And all of them, they will be having some bead-like units on them. Bead-like units will be there, they are called as chromomeres. So chromomeres will be seen in the leptotene and chromosomes will be gradually appearing or visible under the electron microscope. That is the leptotene, which is the first stage. And here condensation of chromosome already it began. Condensation will begin and it will be continued till the end. That is the liptoting stage. About zygotine. In zygotine, what happens is, here you can see two red color ones are there, and red and pink, blue and black are there. They are called as homologous chromosomes. So these two are homologous chromosomes. Homologous chromosomes. Homologous chromosomes means the two chromosomes which are identical in all aspects. You can take the size, shape, genes content, gene order, anything you can take. If they are identical in all aspects, they are called as homologous chromosomes. We have 23 pairs of chromosomes. In that, 22 pairs are homologous chromosomes. Okay, 23rd pair, that may be homologous in females, and in the males it is non-homologous. X and Y will be there, in the females XX will be there. So here the homologous chromosomes means one chromosome is called maternal chromosome out of a pair, and the second one is called paternal chromosome. Maternal chromosome is coming from the mother and paternal chromosome is coming from the father. So the two chromosomes, first pair, that means they will be a pair because they are identical in all aspects. Second pair, again those two will be identical. That type of chromosomes are called as homologous chromosomes. So now you can see red and pink are homologous, blue and black are homologous. Why they have given different colors means to show the next process. So here you can see two homologous chromosomes pairs are there, two pairs of homologous chromosomes are there. So here they are actually different, they will be scattered. But in zygotine what happens is that first pair of homologous chromosomes or second pair of homologous chromosomes, all pairs of homologous chromosomes will come together. They'll pair up. They start pairing together. The process of pairing is called as synapses. They start pairing in zygotine. Pairing occurs. Pairing of homologous chromosomes occurs, which is called as synapses. It happens by some attractive force between them. That attractive force will be called as synapti-enamel complex proteins. Some proteins will be there which will be bringing the two homologous chromosomes from the nucleus from distal ends towards together. They'll come together by the synapti-enamel complex proteins, and after that they'll become a pair. That pair of homologous chromosomes, okay, is called as now bivalent. A pair is called as bivalent. So the end result of the zygotine will be all the homologous chromosomes, they'll start becoming the pairs, and each pair is now called as bivalent. They're called as bivalents, and the process of pairing is called synapses. Synapses or synapses. That bivalent is not that clear in the zygotine because condensation is going on. In the next stage, that is the pachytene, they will be appearing very clearly. So for seeing that particular bivalence, we have to go to the pachytene. They are formed here only, but we can't see them clearly because of lack of that much condensation. So when we come to the pachytene, it will be further condensed. So we can see that bivalence very clearly here. In pachytene, what happens is each chromosome will be having this type of structure. Two chromatids will be there. Like this. One homologous chromosome and another homologous chromosomes. The two chromatids that belong to one chromosome are called sister chromatids. The two chromatids, if they belong to one chromosome, they are called as sister chromatids. So for the white one, the two white chromatids are called sister chromatids. For the yellow one, the two yellow ones are called sister chromatids. But if you take one white and one yellow, these two are called as non-sister chromatids. Non-sister chromatids. So in the pachytene stage, in this, the crossing over occurs. Crossing over is nothing but exchange of genetic material between the two non-sister chromatids of a pair or a bivalent. So here these two, you can see blue one is exchanging the material with the red one. Here also same thing, blue one will be exchanging with the red one and red one will be exchanging with the blue one, like that. Here they have given only one thing, blue one is attached to the red one, and that will happen by some enzymes will be there like recombinase. So blue part will be cut and that will be added to the red one, and red part will be cut, that will be added to the blue one. So exchange of genetic material occurs between always non-sister chromatids of a bivalent. That exchange of genetic material is called as crossing over. Okay. So between the two homologous chromosomes of a pair of a bivalent, the exchange of genetic material occurs by the help of one enzyme called as recombinase, and after that they will exchange their parts, but they are still connected and condensation is happening, it is continuing. That will happen in the pack 18. So you can see here chromosomes are fully synapsed and crossing over begins. That will happen in the pack 18. In the next stage, diplotin. Here what you have to remember is crossing over. Diplotin, what happens is, I gave you here synaptinimal complex proteins are there which are holding the two homologous chromosomes together. That will be dissolved here. The attractive force which is keeping them together will be dissolved. So in the diplotin, as synaptinimal complex starts to disappear, they try to separate away, away from each other. The two chromosomes will try to separate away or move away from each other. Due to that, we can see the X-shaped structures. Here all of you can see X-shaped structures are visible. That X-shaped structures are called chiasmata. You can see clearly here, chiasma is singular, chiasmata plural. Due to the dissolution of synaptinimal complex proteins in between the two chromosomes of a bivalent, they tend to separate. Okay, they try to move away from each other. When they are moving away from each other, already they are having a point of contact. That point of contact or crossing over, they will be looking like X-shaped structures and they are called as chiasma or chiasmata. That chiasmata are visible in the diplotin stage. That will happen in the diplotin and condensation is proceeding. Next stage is diakinesis. In this diakinesis, whatever the chiasmata they are trying to separate, that chiasmata would have reached the ends or poles like this. The chiasmata which started here at the centromere region, they'll move like this and they'll be reaching the ends of the chromosomes. That process is called as, in the diakinesis, terminalization. So diakinesis is characterized by terminalization. Of chiasmata. The chiasmata from the point of origin, they'll move towards the ends of the chromosomes. That is called terminalization, which occurs in the diakinesis. Terminalization of chiasmata, that is the main event. After that, what happens is, in the nucleus, the changes will happen. This nucleolus disappears, nuclear envelope, it breaks here, Golgi ER disappears, and condensation would have been completed by the end of this. So as the nuclear envelope is not there, now the attached bivalents will be released or scattered into the cytoplasm. They will be released into the cytoplasm. So in leptotin, condensation begins and chromosomes appear gradually or gradually visible under the electron microscope with beads called chromomeres. And then in zygotin, homologous chromosomes will pair up by a process called synapsis with the help of synaptinimal complex proteins. The end result will be so many bivalents will be formed. Bivalent means two homologous chromosomes. Pack 18, the two chromosomes of a bivalent will exchange their genetic material by a process called crossing over. That will happen always between the two non-sister chromatids of a bivalent. Then diplotin. In this, dissolution of the synaptinimal complex proteins occur. So they try to move away from each other so that the X-shaped structures, which are the points of crossing over, they are seen very clearly in the diplotin. In diakinesis, that chiasmata from the origin point, they would have migrated towards the ends of the chromosomes. That is called terminalization of chiasmata. And by this time, the chromosomes would have condensed fully, and nuclear envelope, nucleolus, Golgi, ER, all of them disappear. Now these chromosomes will be released into the cytoplasm, and spindle fibers are also formed here only, okay, and they will come and they will attach in the metaphase. You can see in this also the leptotin stage clearly, centromeres are there, and even the chromomeres, you can see the beads are there. These beads are chromomeres, bead like units will be there. In zygotine, you can see two chromosomes are starting pairing up by the synaptinemal complex. You can see that ladder like thing is there. It is bringing them together, that is synaptinemal complex. In the pachytene, between the bivalents, each chromosome is having two chromatids, so it is called as tetrad. One chromosome and another chromosome. For one chromosome we have two chromatids, for two chromosomes we have four chromatids, so it is called as pachytene tetrads. In this crossing over, they exchange their genetic material in pachytene. In diplotene, chiasmata are seen because of the dissolution of the synaptinemal complex. Then in diakinesis, terminalization happened and condensation completed. Now this type of chromosomes are released into the cytoplasm. You can see that centrioles would have migrated towards the opposite poles with the spindle fibers. In metaphase 1, now the spindle fibers will come from the opposite poles and they will be attached, okay, to the kinetochores of the chromosomes. The same event like metaphase of mitosis. So spindle fibers will be coming from both the poles and they will come and attach to the kinetochores of this bivalent. So each bivalent is attached to the two spindle fibers coming from two opposite poles. In mitosis, what happens is one chromosome is attached to both the poles by two spindle fibers. But here each chromosome is attached to only one pole by one spindle fiber because this is the bivalent. And after this, all the bivalents will be placed on the center of the cell. You can see here now. Once the prophase 1 is over, the chromosomes or bivalents are released into the cytoplasm. In metaphase 1, these bivalents are attached to the kinetochores. You can see that red color and green color one and green and red, they have exchanged their paths. Now spindle fibers will come and attach to the kinetochores from both the poles for the bivalent, and then they are brought to the center of the cell. This is the center point of the cell. So they are arranged on the center of the cell. This is called as metaphase plate, to form metaphase plate or equatorial plate. That will happen in the metaphase 1. In anaphase 1, what happens is, out of the bivalent, two chromosomes are there. So one chromosome will move towards one end and another chromosome will move towards the opposite end. So homologous chromosomes get segregated. This is called segregation of homologous chromosomes, separation. Separation of homologous chromosomes of a bivalent occurs in the anaphase 1. Two chromosomes are becoming a pair, no? So out of that, one chromosome will move towards this pole and another chromosome will move towards the opposite pole. Segregation of homologous chromosomes occur in anaphase 1. Then in telophase 1, these chromosomes which have moved towards the poles, okay, they will be decondensing into chromatin. But they will not reach to the too much extended state, okay, but they will be becoming the chromatin. So by the end of the telophase 1, you know telophase is exactly opposite to the prophase. So by the end of the telophase 1, we'll be having the nuclear envelope formation, nucleolus formation, Golgi formation, ER formation will be there. So one nucleus will be dividing by the end of the telophase 1 into two daughter nuclei. Imagine here you can see 4 chromosomes we have. By the end of the meiosis 1, particularly after telophase 1, you know here cell wall will be formed or the furrow will be formed, cell plate will be formed by cytokinesis, and it will divide this into two daughter cells. These two daughter cells are called dyads of cells. Two daughter cells are called dyads of cells. So here we have 4 chromosomes. Here all of you can see only 2 chromosomes are there in each daughter cell. So here 2n is equal to 4. Here n is equal to 2. We have started in the cell with 4 chromosomes. Now after the meiosis I, each daughter cell is having only 2 chromosomes. That is why this meiosis I is called as reductional division. Reductional division. And in the interkinesis, okay, they will be reaching like extension phase, but they will not be fully extended like the interphase. Okay, and the cell will be preparing itself without any DNA replication to undergo the meiosis II. So what point you have to remember here is, in anaphase I, there is no splitting of centromere, no splitting of centromere like mitotic anaphase. Here segregation of homologous chromosomes is happening. Segregation means separation of homologous chromosomes of a bivalent. That will happen in the anaphase I. No centromere splitting. Metaphase means just all the chromosomes of the cell will be brought to the center to form the metaphase plate. Segregation of homologous chromosomes to the respective poles. Telophase, formation of 2 daughter nuclei. Chromatin formation, nuclear envelope, nucleolus, Golgi ER formation. Then cytokinesis will happen. If it is an animal cell, it occurs by furrow. If it is a plant cell, it occurs by cell plate method. And one cell will be dividing into 2 daughter cells by the end of the meiosis I. Both the cells are exactly having half the number of chromosomes to that of the parent cell. Exactly half. So meiosis I is called as reductional division. I hope all of you understood. Yeah, cell plate. Yes, cell plate means in the plant cell, yesterday I have told you Golgi complex will be accumulating in the center and it will be growing towards the site. Okay, centrifugal manner. Yes, that is called cell plate. It divides one cell into 2 daughter cells. Yeah, any doubts more? If you have any doubts, you can ask. Any doubts? About what is the use of what DNA in plants? Use of DNA is a genetic material, without which the cell is unable to carry out the hereditary functions. DNA is a master molecule because it controls all activities of the cell. All living organisms will be having either DNA or RNA as a genetic material that will have genes which will code for all our characters. What is the meaning of homologous chromosomes? Yeah, a pair of chromosomes which are identical in all aspects like size, shape, structure, genes, gene content, gene order, they are called as homologous chromosomes. You have 23 pairs of chromosomes, no? 23 you are getting from your mother and other 23 you are getting from your father. Once again explain metaphase I. Okay. In metaphase I, the bivalents will be placed. Bivalent means a pair of homologous chromosomes. That bivalence will be placed in the center of the cell by the spindle fibers. So that is called as metaphase plate or equatorial plate. It is required for the proper distribution of chromosomes to the daughter cells. Does chromatid number double in meiosis? Chromatid number, is it doubling? The chromatid number doubles in the S phase only. What here I want to tell you is chromosome number is reduced to half. Chromosome number is reduced to half. Okay. If you take the S phase and G2 phase of interphase, chromosome number is constant, but the chromatid numbers increased. Yes, any further doubts more? Yeah, 46 we have. So 23 you are getting from your mom and 23 you are getting from your father. So that 23 together, 23 pairs we will say. So 23 pairs of homologous chromosomes we humans have. Okay, Anunaya. Okay. Any further doubts? Yeah, ma'am, doubt Mallikarjuna. Okay, what is your doubt? Balu, what is your doubt? Ask the doubt. 22 pairs of autosomes and 1 pair of allosomes. Yeah, 6 chromosomes. Yes, correct, Veda. Right. Autosomes and allosomes, correct. Plants don't have brain like us, then how do they live? They don't need brain to live because they don't have any systems. They don't have any systems. Only systems which are present in the plants are root system and shoot system. Without brain also they can live. Okay, that's why they are not complaining though we are plucking all the fruits, leaves, flowers, everything. Without centrosomes, how can the spindle fibers form in a plant cell regarding? Okay. They don't have spindle fibers. I mean, centrioles will not be forming the spindle fibers here, but they have in plant cells microtubule organizing committees will be there. MTOC they are called as. Microtubule organizing, okay, center that will be giving rise to the spindle fibers in the plants. What is synaptinimal complex? Yeah, it is made up of proteins. It is made up of different proteins that will be holding the two homologous chromosomes together, to hold them together. You have seen in this picture like a ladder. All of you see here, the ladder like thing is there. This is the synaptinimal complex. They are made up of different proteins and they will be having the charges positive, okay, negative. So they will be holding them together. Yeah. Any further doubts? Yeah. Synaptinimal complex means it is made up of proteins to hold the two homologous chromosomes together. Okay. That's all. Shall I ask you questions? 2 questions? No doubts. Very good. So all of you are attentive. Garuda. Okay. No doubts. Transportation system in and photosynthesis and also reproduce, then it needs brain to do these things. Okay. No brain is required for the plants to do all those processes because photosynthesis is done by the light trapped by the chlorophyll pigments which are present in the chloroplasts. They are not highly evolved like us, animals. Yes, cytokinesis. Yeah, cytoplasmic division. Cytoplasmic division. It occurs by cell plate method or the furrow method, depending on the cell. Furrow method means in animal cell, cell plate method means plant cells. Okay, understood all of you? Shall I ask you questions now? 2 questions. Okay. Now, in which phase chromosomes gradually visible? In which phase chromosomes gradually visible? The options are leptotene, zygotene, pachytene, diplotene, diakinesis. First one, yes, Garuda. Correct. Leptotene, yes. Okay, good. In which stage of prophase 1? Yes, in which stage of prophase 1 crossing over occurs? Crossing over. In which stage of prophase 1 crossing over occurs? Leptotene, zygotene, pachytene, diplotene, diakinesis. Which one? Very good, Pac-18. Tikshina, very good, Pac-18, Pac-18. Right. Now, in which stage bivalents are formed? Bivalents. In which stage bivalents are formed? Leptotene, zygotene, Pac-18, diplotene, diacansis. Yes. Darshika, yeah, tell me. My name, okay. Geeta, I'm Geeta. Very good, Tikshana, zygotene. Bivalents are formed in the zygotene. Bivalents. Afifa, wrong. Bivalents are formed in zygotene. Okay. Chiasmata will appear in which stage? Chiasmata. Chiasmata. Options are same. Leptotene, zygotene, Pac-18, diplotene, diacansis. Where can you see chiasmata? Zygotena? No? Very good, Bunny. Fourth one, very good. Diplotene, Madhumita. Very good, correct. Tikshana, right. Good. Now, among meiosis I and meiosis II, which is called reductional division? Which is called reductional division? Meiosis I or meiosis II? Tikshana, 1, very good. Last question. If one cell is having 46 chromosomes, it enters into meiosis I. After meiosis I, how many chromosomes are present in the daughter cells? After meiosis I, how many chromosomes are present in each daughter cell? Yes, Tikshana, very good, 23. Very good, 23. In each daughter cell, we'll be having 23. Very good. This cell ploidy, is it diploid or haploid? What is a ploidy? If 2n is equal to 46, then 23 means what is it? N or 2n or 3n? It is n. N is equal to 23. Yes. Okay. I hope all of you understood. Prepare this and come tomorrow. Let us see the last phase, that is meiosis II. I hope all of you understood. Give a thumbs up if you understood. Yes? Okay. Okay, ma. See you tomorrow. Bye. Thank you.
Good morning, students. Good morning, students. How are you? Hello, everyone. How are you? Krishna, Farheen, Sita, Guraju, Sairaj, Butterfly, Madhumita, Lekhya, Harshit. I'm good, Harshit. How are you? Fine. That's good. That's good. So in previous class, we learnt about common human diseases or health conditions. We learnt about infectious pathogenic diseases which are communicable and non-infectious, non-pathogenic, non-communicable diseases also, right? So now, today, we will be knowing few points about, we will be knowing a very brief concept of most interesting part of your body, most advanced organ of your body, the organ or a part of your body which controls every other organs, and this itself is almost 80% of lipid fat content of your body, and this organ itself consumes at least 20% of inhaled oxygen, and cells in this organ do not divide usually. Cells in these organs are almost like copper wires helping in conduction, and cells in this organ are considered as one of the longest cells in animal kingdom. So what was this mysterious yet interesting organ? Tell me. Yes, brain. Brain. So listen to this properly, you will understand. Follow me properly, you will understand. Don't split screens and share your time between other apps and classes. That was the reason why you are not able to follow properly. Please, I'm telling you again, focus on one aspect. You will understand. I'm repeating thrice, 4 times, 5 times also, just to make you understand simple concepts, because as such you cannot start your 11th standard all of a sudden. No, you need some basics before you jump into those specialized chapters. That's why. Okay. Now, brain, most interesting and increase your volume, Martha, first. Check your network settings, change your audio settings. That will make it more and more visible and more clearly, much clearly audible also, right? Done? Okay. So brain, most interesting part of your body, and this part of your body controls every other organ also, and your brain mainly consists of 2 types of cells. Some cells help in memorization, impulse conduction, getting stimulus, giving responses, whereas some other groups of cells were there which actually help, which actually provide food, which actually remove debris and ammonia from your brain. The main type of cells of your brain which are actually memorizing things, understanding things, receiving stimulus, sending responses, and responsible cells which are responsible for thinking. Those are neurons, nerve cells. Okay. And neurons are considered as one of the longest cells of your body also. Giraffe neck has very long neuron, at least 10 meters. Gigantic squids also possess very, very long neurons. In your body also there is a long neuron here. Yeah. Whereas there are some cells which were between neurons, like a gum, glue, and these cells help neurons by giving them food, providing them some matrix, clearing some debris, ammonia, and if possible, they may also provide protection to neurons. Those additional cells which help neurons are called as glial cells, glia. So some books also mention that as neuroglia. So brain consists of 30 to 40% of neurons and 70 to 80% at least of neuroglial cells. Okay, so neuron and glia. All of us are aware of neuron, right? Neuron, can you tell me some of the components of neuron? A neuron may have a cell body, a very long elongated axon, which ends in nerve terminals, right? So this cell body has tiny hair-like structures with which it actually receives impulses. These extensions of cell membrane of cell body, we call them as dendrites. Dendro means branches. Dendro means branches. And it consists of a single nucleus at the center. Okay. There was dense cytoplasm, whereas in cell body we can see a peculiar thing: ribosomes and rough endoplasmic reticulum. They join to form blackish grayish color structures like this. Rough endoplasmic reticulum and ribosomes, they associate with each other to form blackish color or to grayish color looking bodies. So these dark colored granules, dark colored bodies are referred as Nissel's granules or Nissel's bodies. Yes, Nissel granule is nothing but combination of rough endoplasmic reticulum with ribosomes. Which kind of ribosomes? ATS ribosomes. Okay, and coming below, this elongated portion of neuron was called as axon. Done. And often axon might befriend special glial cells, and those glial cells help in formation of fatty layer around axons, like Schwann cells. Schwann cells come near axon they befriend, they support axon like this. Slowly, Schwann cell will form a coating of fat substance around axon like this. Like this. The coating of fat substance. The coating of fat substance was known as myelin sheath. Myelin sheath. Done. Now for your information, if you look a neuron under microscope, this cell body may look grayish in appearance due to presence of nissl bodies. Okay, but axon with its myelin sheath may appear white or yellowish like butter-like color because of myelin sheath. Grayness is due to nissl granules and yellowishness is because of myelin sheath. So gray part of neuron, yellow part of neuron. Done. These are the cells which were mainly responsible for receiving stimulus, sending responses, memory storage, thinking, analysis. Like, now, if anything happens to brain, any damage, physical or physiological, so it may lead to some disturbance in brain functioning, that should be immediately resolved. What is the discipline of medicine, branch of medicine, which deals with study of brain, related disorders, and possible treatments for brain? Tell me, what is name of that medicinal field which deals with study of brain, its disorders, and possible treatments for brain? Yes, very good. Neurology. The doctor who does this was neurologist. Neurology, very good. One of the most fascinating fields of medicine was neurology. Okay, now this is how brain looks inside of your body. See? So you can see brain has a slight pinkish gray appearance. Pinkish gray appearance. That's how this is a side view of your brain. And if you see side view, front view, back view. This is back. Guys, what is back in scientific language? Back, what is back? Ventral or dorsal? Back is dorsal. This is side view. This is ventral, front view. So back, dorsal view. Side, we call that as lateral view. Front, ventral view. So this, so side, back, front. So to make it much more simpler, our physiologist has did a very great job. They have split brain into three components. What are those components? First one, forebrain. The first component was forebrain. Forebrain. Just below forebrain, you can see midbrain, and just below midbrain, you can see hindbrain. Guys, this is front. This is back. What is synonym for front? Ventral. Synonym for back? Dorsal. Okay, so three parts: forebrain, midbrain, and hindbrain. Done. This is back, dorsal. This is front, ventral. Okay, so forebrain, midbrain, hindbrain. Here you can see hindbrain inside this part. Forebrain was here almost, that is. Okay, three parts: forebrain, midbrain, hindbrain. And there are also technical terms for these parts of brain. Forebrain was technically referred as prosencephalon. Prosencephalon is forebrain. Midbrain was technically referred as mesencephalon. Mesencephalon. Whereas hindbrain was referred as rhombencephalon. So we can see, we can observe a peculiarity here. Encephalon. N inside cephalus, head. The first part of brain you see from outside was prosencephalon. The part of brain which is behind your body, back, hind, rhombencephalon. The part of brain which is between prosencephalon and rhombencephalon is middle, mesencephalon. Done. So forebrain, midbrain, hindbrain. Prosencephalon, mesencephalon, rhombencephalon. Okay, got this till now? And for information, entire brain was actually covered by special layers to protect them. Entire brain was actually covered by special membranous layers to protect it. So there are three membranes. Membrane found attached to brain itself, innermost membrane, and a membrane which was outside of the brain, which is actually attached to cranium, and there was a membrane between outer membrane and inner, like this. So these three protective layers were referred as meninges. Guys, we do mammals possess 3 meninges, but do not expect same 3 layers in every animal in animal kingdom. If you ask about fishes, any fish brain is simple and it was only covered by a single meninge. It was referred as meninges primitiva, primitive meninge. And lucky enough that we were mammals, our brain got covered by 3 meninge layers. Outermost, the hardest meninge, which was attached to cranium, was called as dura mater. Dura means durable, hard. Dura mater. Innermost, thinnest and softest meninge, which is actually touching brain also. This was pia mater. Observe the spelling, it's not matter, it's mater. Mater means mother, maternal, paternal. That was the spelling, not matter, pia mater, dura mater. No, don't pronounce like that. Pia mater, dura mater. And between pia mater and dura mater, there was a layer. This layer looks like, in microscope, under microscope, it looks like a spider's web filled with blood vessels, some cells. Because of this web-like appearance, this middle layer was named after spider. Therefore, arachnoid layer. Arachnoid membrane. Arachna means spiders. So inside to outside, inside to outside, pia mater, arachnoid membrane and dura mater. Inside to outside, pia mater, arachnoid membrane and dura mater. The innermost meninge, pia mater. Outermost meninge, dura mater. And meninge, which has spider's web-like appearance and was found inside, below dura mater, above pia mater, was arachnoid membrane. Which meninge touches brain? Pia mater, innermost. Which meninge touches cranial bones? Dura mater, hardest, outermost. Like this. A question may come asking you arrangement of meninges from inside to outside or outside to inside also. Both are possible. And these meninges have functions. They protect your brain from shocks. They are like shock absorbers mainly. They do not allow leakage of any fluid out of your brain, like a cerebrospinal fluid. Cerebrospinal fluid, CSF, is almost like a plasma. Nothing new in it. I will tell you details about CSF once you get into the subject. Okay. Pia mater, arachnoid membrane, dura mater. Okay. Yes, we'll get into CSF, not now. I will tell you importance of CSF once you complete the part. And the visible part of brain, what you see, it has many folds, that is forebrain. Guys, forebrain is actually visible part of your brain which looks pinkish gray in color, and forebrain has got two components. It has got cerebral cortex, which is actual pinkish gray, visible, and an invisible part known as diencephalon. This is visible part of forebrain. This is invisible part of forebrain. So cortex. Cerebral cortex could be split into two parts. Left hemisphere. See, cerebral cortex can be split into two parts. Left hemisphere, right hemisphere. Left cortex, right cortex. There was a line, sulcus. See, left and right. From front view, left, right. Okay. And surface of this cortex looks grayish in color, pinkish gray. Pinkish appearance is because of blood vessels. Grayish appearance is due to arrangement of cell bodies here. Like this. And cerebral cortex or cerebrum. So as I told you, the visible part, the cerebral cortex is indeed also referred as cerebrum. So cerebrum has two hemispheres, left hemisphere and right hemisphere. And guys, for your interesting aspect, left brain controls right side of your body, right brain controls left side of your body. Zigzag, it zigzags in your head region. Left brain controls right body, right brain controls left body. Now, if you are a right-handed person, which means that if you're brushing with right hand, if you're handling with right hand, writing with right hand, lending right hand every time, you are using right body, which means you are left dominated. Same time, if you're using left hand to write, lend hand, use it more, you're lefty, which means you are right dominated person. So zigzag. If you're right-handed person, left is much better working for you. If you're left-handed person, right is better working for you. And in between these hemispheres, see, there was a thick tuft of nerve fibers connecting both these hemispheres. Inside between these hemispheres, you can see a dark region. There was a thick network of nerve fibers connecting left hemisphere to right hemisphere. Very, very important part of your brain. So this bridge-like tissue which connects left hemisphere to right hemisphere, it was referred as corpus callosum. Is somebody asking me? Yes, Yashu, we can use both parts of the brain, but not equally, because the regions or areas on one side are not exactly same as the other side. Pain, happy emotions, sad moments, excitement, fear, or all these emotions are not in one side only. They're distributed unevenly. Brain was not even organ. Brain has no same structures physiologically. No. For him, sire, that was not corpus luteum or corpus luteum. That was corpus callosum. Corpus callosum. Corpus luteum will be in females when they menstruate. For him. So, a tissue which is between left and right hemisphere is corpus callosum. And brain cortex may look grayish outside, but interior brain is actually yellowish. Now, I have a question for you. Why cortex looks grayish in color? Reason, please. Why cortex appears grayish in color? Because of concentration of cell bodies. And cell body, why do they appear grayish in color? Because of presence of nasal bodies. Yes, grayish. Pinky, okay, blood vessels pinkish, but grayish because of concentration of cell bodies. Therefore, the outer part of brain was usually referred as gray matter. If you look at interior part of brain, that may look bit yellowish to whitish. Why? Inner part of cerebral cortex have yellowish or whitish appearance. Any reason? Why? Interior location of cerebral cortex, inner part of cortex has whitish, creamish, yellowish appearance. Any reason? Yes, because of axons having myelin sheath. Therefore, interior was referred as white matter. Guys, you'll understand gray matter and white matter only when you know importance of neuron. So cortex surface has richness of cell bodies, cortex interior has richness of myelin sheaths. Therefore, cortex appear gray because of cell bodies and inner parts of cortex appear yellowish because of myelin sheaths. Done. And so forebrain, this part, we can see it was highly folded. If there are many folds, folding will increase surface area so that they can accommodate more neurons. Even in these foldings, there are elevations and ridges, elevations and depressions, elevations and depressions. Elevations are known as gyrus, singular, gyri, plural, and depressions are referred as sulcus, singular, sulci, plural. Sulcus, gyri, sulci. Gyri elevation, sulci depression. Gyri elevation, sulci depression. So because of gyri and sulci, there might be more surface area and brain can accommodate more neurons. Done. So forebrain has special regions to receive stimulus. We call them as sensory areas. Special regions to send responses called as motor areas. And there is a peculiar part of brain having associations that was neither completely sensory nor completely motor. We call them as association areas. The main information storage, processing, analysis, intelligence part of your brain was forebrain, cerebral cortex or cerebrum. So cerebrum stores information. Cerebrum is largest part of your brain. Cerebrum is visible part of your brain. Cerebrum pinkish gray in color. Cerebrum stores memories. Cerebrum is main processing part of your brain. Done. And just below cerebrum you can see invisible part of forebrain also. This invisible part of forebrain is called as diencephalon. Guys, diencephalon has got three parts. It has got middle part is referred as thalamus. On above, epithalamus. And below it was hypothalamus. We may discuss about epithalamus and hypothalamus in the context of hormones later. As of now, just memorize that simply. Diencephalon is part of forebrain. It consists of three components: thalamus, epithalamus, hypothalamus. That's it. Okay. And just below forebrain, there is a midbrain, right? Midbrain has bulb-like structures called as colliculi, and there are four colliculi. Therefore, corpora quadrigemina. Two collicula help in better vision, optic lobes, and two collicula help in better hearing, auditory sense. Therefore, midbrain helps you to see and listen better. Midbrain helps you to look at good videos and listen to good audios. Therefore, video and audio lobe. That is midbrain. Whereas hindbrain has three parts. This is pons varoli, this is cerebellum, and this was medulla oblongata. Hindbrain has three components: pons, cerebellum, medulla oblongata. Pons, cerebellum, medulla oblongata. Three parts: pons, cerebellum, medulla oblongata. Three parts in hindbrain. So pons is responsible for passage, or it acts as a bridge between main brain part and other neurons. And pons also controls respiration. Whereas cerebellum is a part of your brain which looks like cerebrum, which has also got two hemispheres halves, and cerebellum controls how you balance your body. Cerebellum controls your pronunciation. Cerebellum controls fastness of your speech, how fast you speak. Cerebellum controls your grip over language. The first body part which will be damaged if you drink alcohol was obviously cerebellum. An alcoholic have disturbance in cerebellum, therefore they cannot walk properly, they cannot speak properly, they do not know how to balance their body. That's why police warn them, don't drink and drive, because you don't know how to balance. They lost equilibrium. Okay? And the last part, medulla oblongata. Medulla oblongata controls reflex actions. It controls your respiration. It controls acids of your body. It controls heartbeat also. So just I'm telling you details. Let us not get into depth of the subject. Once you come here, I'll let you know detailed anatomical structures of brain and even more and more details of brain. Okay, as of now, memorize forebrain, midbrain, hindbrain, cortex, diencephalon, audio video center, pons, cerebellum, oblongata. That is enough. Okay, do not get into depth of details. The more you learn about brain, if you learn about brain more, there is one advantage and disadvantage. The more you learn, there is more unlearned things, because information about brain never ends. There is everyday new thing getting discovered in brain, so you should keep on updating. Less you learn, you become ignorant. So better to stick to what I say. Just stick to basic things, okay, fundamental things, okay. We'll see. So we'll end here for today. We'll meet tomorrow. Thank you. Stay home, stay hydrated. We'll meet you soon tomorrow. Done? Bye.